tell from the shiny text back here, I'm going to give a seminar/workshop about genes and genetic technology and basically stuff I find really cool. Okay, we'll start off with the basics of transcription and translation. Then we'll talk about some interesting things like Hox genes. We'll be talking about the FOXP2 gene. We'll be talking about epigenetic regulation a little bit. And then we'll be doing a live demonstration on precipitating DNA from a solution. Yay! That's going to be my indicator for my slide web. Yay! Also more jazz hands, thanks. So, why do I do genetics? And why do I want to talk about it? First, the more I look into it, the more I think that some gene therapy is the future of medicine. Next, it's like super relevant because we're made up of the stuff. If you haven't learned already. It is also incredibly pretty, and to prove this to you, I'm going to show you a video that my biology teacher from college showed me, and it got me so interested in microbiology and genetics. I thought I'd show you guys. And again, I'll open my phone to check that it is good. Okay, so just for curiosity's sake, how many people can tell what's going on in that video? Like, any part of that video? Call it out loud. Anyone?
Actin-myosin interaction?
Anyone else?
What's to that level then?
No, keep going. Anything is correct.
I kind of have a vague idea that, like, you've got bonds forming and everybody's losing electrons, and as a all the bonds are breaking.
There's a lot of DNA replication.
Okay.
There was translation.
Yes.
So much pop.
Yes.
Anyone mention something? basically the giant netty thing that was everywhere. There were
The Golgi body was that.
the Golgi body, that gloopy looking thing that was in like all
Pancakes.
That's what I remember. It. Pancakes. There was a mitochondria, that thing that was kind of looked a little phallic.
There wasn't a double helix. I didn't see a double helix.
There wasn't a double helix because double-stranded DNA isn't actually featured in that video. It's just, if cell spawn is a thing, that's it. Basically. That's the sexy stuff. Alright. It is awesome. Keep going. Alright, now. No. Just hit the back arrow. No. Arrow keys. Stop pressing forward. Okay. I'm going to draw for you the basic cells because I think it's important to note where in the eukaryotic cells, which are animal and plant cells, DNA expression is located, which is also a nice convenient segue. Alright, so tell me if you guys cannot see this, and this is like stupidly basic, doesn't contain everything. Cell wall made up of a phospholipid bilayer, so one side hates water and the other side, which is facing outside, really likes water. Now, here's the nucleus. The nucleus is surrounded by a nuclear envelope, which joins into this thing called the endoplasmic reticulum, which no, you don't have to remember. We call it the ER. Yep. There is a rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough side is closer to the nucleus, and that contains tons of these things that we call ribosomes. We will get to the ribosomes soon. So you have the rough ER and the smooth ER. This is where all the DNA is kept. Now you'll notice I haven't drawn them in the classical shape of chromosomes. That's because they only really become chromosomes during cell replication. Then, replication. It usually just stays in this big messy spaghetti, like scribble, basically. In here is where transcription happens. That is DNA to RNA. It goes out of the nucleus into the endoplasmic reticulum as mRNA, then it undergoes translation. So that is the mRNA to a protein. That is all we will cover in terms of transcription and translation. There are tons of other organelles out here, little cell organs, that package and add stuff onto the molecule before it is actually utilized. However, that is not covered in this. Any questions before I continue? Cool? Cool. Feel free to interject at any point if I've either gotten something wrong or you don't understand anything. Because as I said before, I hate the term speeches. It freaks me out. This is basically just a giant conversation. Yay! Okay, so DNA structure. This is the double helix. It contains a sugar phosphate backbone and nucleotide bases. I'm going to go to the other side so you can see. Okay, so both sides run anti-parallel to each other. They match up from 5' to 3'. The 5' end is the one that contains the phosphate. The 3' end is the one that contains the sugar.
The hydroxyl.
Yeah, hydroxyl and sugar. So this is the 5', this is the 3', and so on the other side, as they're running anti-parallel, this would be 3', and that would be 5'. Okay. Nucleotide bases. We know them as A, T, G, and C. Does anyone know the proper names of these, apart from these two? Thymine. Yes. Excellent. Who knows what pairs up with who?
A, C, B, G.
Yes. Okay. They're all bonded together by hydrogen bonds, then they all wind up to form a double helix. From that, it coils further. It coils further and further and further until what they're known as supercoiling. Have you ever like 12 pieces of string and it gets 12 up so tight that it sort of bends in on itself? That's basically what supercoiling is. And then once it's supercoiled up tightly enough, it binds around the scaffold, as you can see there, which then winds around again around something called a histone protein, which is not actually pictured here, so I've neglected to put that in. But basically, it winds around these spools known as histones, they clump together, then it winds around again into these major, basically large structured coils here into a chromosome. Can anyone explain to me why it is beneficial for it to wind up so tightly?
It's really small.
Apart from you two.
It's really small?
Yes, that is one of the many reasons, because we have enough like DNA in a cell, isn't it, to like cover a football field? or something? It's an obscene amount of genetic data within our DNA. Anyone else? Can be anything. Any answer is correct. Well, not any answer, but any suggestion is a good one.
Maybe it's stronger.
Almost there.
So they're not entangling with each other?
Yeah,
And they want to split?
pretty much. You get a prize of some description eventually.
Information.
I'll give you a wedgie.
Doesn't pierce the.
It's also really helpful in the sense that not only can it like not tangle
Krisha
up with
has a
anything else.
suggestion as well.
Oh yeah, what is it?
Krisha's suggestion.
You said
It
they
doesn't.
said.
It doesn't pierce the walls of nucleus.
No, that's not correct, but I like your suggestion. Okay, another benefit to having it so widely tightened is so that it doesn't mutate as easily. One of the main reasons it's like bundled up together is so nothing can sort of get in and break a bond or replace a base, just because there's so much steric hindrance by everything else going on within that supercoiled structure.
It's like wrapping molecules in cotton wool.
Yeah. Okay. Transcription time. Transcription, as I said, DNA to RNA, it happens inside the nucleus, and it is always from 5' to 3'.
And why
Now.
do we have RNA? Why do we have
I
RNA?
do
What
believe
is
she's
it?
about to say
Answer.
that.
Okay. Actually, that's a good question. Why do we have RNA?
Quickly, what is RNA?
RNA.
Like the difference.
Okay, DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. One of the reasons why it goes from DNA to RNA to protein instead of DNA to protein is because there is a lot, among other things, there is a lot of processing that happens in between the transcription and translation, but you do not want to lose that original blueprint. For example, genes can get spliced, different methylation of genes. Do you want to add anything to that? Because I'm obviously forgetting something.
Well, the main difference between ribonucleic acid and deoxyribonucleic acid is that the RNA is less stable.
Mm-hmm.
So basically, you're making, it's like making a copy of a stone tablet using paper, so that you can use that copy to do all kinds of crap and not worry about if it's broken or lost or anything, because you still got the stone.
Yeah.
It's
Isn't
also
the main difference that like RNA is one?
It can be. It isn't always.
It's not always.
I'll explain that a little later.
in humans.
RNA interferes.
Yeah, do genetics, go.
Okay. Another main difference, which I failed to mention earlier, is that the pairs of DNA are A, T, G, and C. The pairs of RNA are A, U, G, C. And the reason is it's part of the reason why RNA is less stable. U is far more likely to be not methylated, but
U is basically completely different from the rest of the bases, whereas if cytosine C is attacked by a particular molecule, it will become something that to the cell looks like T.
yes.
You end up with all these transcription errors, whereas U looks completely different.
Okay, so this is really blurred, but basically, the DNA unwinds itself to a point where this macromolecule comes in, and it's basically a combining of many transcription factors and many RNA polymerase into this giant sort of superstructure, and it wedges itself in between the two. Basically goes from 5' to 3', the direction of transcription, and starts adding in RNA nucleotides, which are just single bases corresponding to the template strand here. At the very end, it will detach itself. The superstructure will detach itself as well as the completed RNA molecule, which we'll know as messenger RNA or mRNA, and that will leave the nucleus. Now, this is This is like the super simplified version, simply because there are like at least 7 or 8 different molecules involved in transcription as well as translation. But I'd rather it be about how like each thing works rather than you memorizing a bunch of names. Okay. Has anyone got any questions about this before I continue? Yes.
Sorry, so this is transcription is part of this wide process of DNA replication.
Yes. Well, not replication.
Right, okay.
Replication is completely different because it sort of splits into, and then I always think of it as a pair of pants because it like splits into and then starts duplicating into.
Paragene.
Yes.
Fantastic.
Sorry, I love it somehow. What is expression?
It's basically taking the gene that the information that is transcribed in DNA and turning that into a protein.
Okay.
That protein like has huge implications to numerous metabolic pathways, but it's always like a protein. It's not like this is the skin snake. It's yeah. Translation. Outside the cell nucleus, we've gone into the endoplasmic reticulum, as I told you. The raphia has all of the ribosomes. Ribosomes are really freaking cool, and I'm gonna start drawing now. Okay, so.
Sorry, maybe put ahead of
Have a good evening.
The noise ribosomes make.
Yes. Okay, this is my half-formed mRNA, because I'm lazy and I don't need to wait. This is what's known as the XSAC sequence. It is basically telling the ribosome unit to bind here. This is where you want to be, because if it moves even one base pair, there's something called a frame shift, and it basically misreads everything, which has very large implications, particularly if it's at the start of the molecule. Okay, so, ribosome. There is a small subunit and large subunit. It looks kind of like that. There are 3 regions. We call them the E, the P, and the A.
Environmental
EA.
Environmental Protection Agency?
Yes, we call it the Environmental Protection Agency.
That's what happens.
Translation starts when one tRNA, which is like this. This is the actual tRNA here. This is the amino acid. So, another tRNA molecule comes into the A site with its ribosome. It moves along one. So let's pretend that this is where it's moved along to, because I haven't drawn enough. And so it's done a shift to one frame. These two join together. The empty tRNA leaves it, ready for another one to come in. So it adds one, shift one, remove one, add one, shift one, remove one. Now the reason why I've grouped 3 base pairs together is because they are known as codons, and there is a fancy looking table right down there, which is blurry to everyone else except for me, that basically has the sort of genetic, well, the codon code. And it's basically every 3 base pairs corresponds to 1 amino acid. And you can see, I'm not sure if you can see actually, but there might be 4 different variations or so for 1 amino acid. Now, why do you think that would be?
Safety. In case, it moves or something, and it's, or like there's a little mutation, if it's got more than 1 code for it, then it'll be easy to actually not mutate and
Yeah, basically. Like another word for this is redundancy. If you have like many things coding for the one thing, if there is a point mutation, so one base pair changes, you're far more likely to have a silent mutation where nothing major changes.
Just quickly, so therefore, in terms of actually producing the amino acid at that point, it's only the first 2 that actually matter. The third one is just there as a placeholder to pull it over and the next one to be made.
Well, no, it's not really, because in many cases,
That's a general rule.
yeah.
Generally, it's the first 2 that matter, but not always.
So now over here with the highlighted ones, they are also very important because although we have the XSAC sequence at the very beginning, like I'm not sure if you remember that, that said where the ribosome is bound, it is important that you have a start codon and a stop codon so you actually start and stop translation in the appropriate place. Yeah?
Is it possible for something to just not have a stop codon and just keep going?
Yes, it is. But, you know, whether it's viable or whether it's good is debatable. Probably not. But yes, you certainly can have instances where for some reason the stop codon is mutated and it doesn't work anymore. For example, UGA could quite easily become UGC. In many cases, after the actual gene is after the codon, like it will just run to the end of the mRNA, and usually it'll just have like erroneous, I think it's just erroneous bases at the end, which could have sort of implications for how the protein ends up folding, and therefore its functionality heavily depends on the gene, heavily depends on the protein. So yes, it is possible. Is it good?
It would usually be lethal.
Yeah. Okay. Fox genes. Now we're on to the cool things. Fox genes. They are in all, if not most, multicellular organisms. They're in just everything, but they're seen mostly when you're going from an embryo to a full-blown, like, baby child. Because they're mostly developmental. It's like if you were to compartmentalize the body and each part had like a certain Hox gene dictating what it became. So this would be like the head Hox gene, 2 eyes right here. This would be the, rump Hox gene, and it would dictate if I had a tail or not. Now, some of the really cool things about it is that it is so conserved. It's one of those things that is never like changed because you only use it during the developmental stage. You use it once during while you were an embryo basically, and you basically don't touch it throughout the rest of it because everything's where it should be. So it's just tightly wound around these histoproteins and nothing touches it. So for example, if I had a fly and chicken, I wanted to breed a fly with chicken wing Hox genes. The fly would still live, the fly would still function exactly as it was before, just because it is so conserved. It would basically be like, that has 2 wings, that has 2 wings, okay, go. There are some interesting things that do arise if you lose Hox genes or a Hox gene is, for whatever reason, very mutated. And there's an example right here, which you can't really see very well. But there are fetal Hox genes for Drosophila melanogaster, also known as the fruit fly. These are the normal antennae that you'll see. This one has been either lost or mutated, and they were like, we'll put 2 feet here. So now they have like 2 little feet growing out of their head. But there have also been other known fruit fly mutations within the Hox gene, which basically, like there's one that's from the labial region, which controls placement of the mouth and the anus. And basically the organs are there, but it can't there's no mouth and there's no butt, basically. So Hox genes are very, very relevant, obviously. And one of the very cool things about it is that if you were to track where the Hox genes are and where the self-expressing these Hox genes are located throughout embryonic development, you can see that different species have different patterns which might indicate one common ancestor. And I could go into a whole rant about that, but we might not have time.
Can I just say one thing?
Yes.
One of the coolest things about Hox genes is if you have a duplication in one, it can change basically the amount of that organ you have. For example, giraffes have a whole heap of duplicates of the neck gene. And they basically just end up with this long neck.
Yeah.
Same with ducks.
So it's basically like head gene, neck, torso.
Ducks, they have the neck gene. It's quite long.
Yeah, and same with snakes, they have like a huge sort of abdomen/tail.
And no leg gene.
No leg gene. So basically, if I wanted an extra arm, I'd have to clone myself and add another arm gene in there, and hopefully
could, it's just not doing it.
It'd be very painful.
Yeah, it would A, be very painful, and B, would be really hard and probably very expensive. So, I mean, say goodbye to wings would be God. Okay, next. Fox P2, another cool thing. Okay, this is a gene that is correlated with not only pattern recognition, but sort of mouth dexterity and language. I bring this up because there is a family that has been noted to have a speech disorder that is genetic and hereditary. It is a mutation within the FOXP2 gene. It not helps with. It hinders the ability to write. It hinders the ability to pick up on chromatic patterns, and it makes it really hard to focus on like facial movement and to speak clearly and to sort of engage in a proper grammatically correct conversation with someone. It's not that they can't, it's just that it's a lot harder to. So gene mapping has revealed that all patients have a missense mutation in the FOXP2 gene. Who knows what a missense mutation is? And can they explain it to me apart from you two? You had your hand up?
Is that when the codon change to, not the codon, the base is changed to a different one rather than being chopped out?
Yeah, a missense mutation is a base, nucleotide base, changing to a different one, which then results in a potential difference in codon. In this case, because it is a missense mutation and not silent, it would result in a different codon. The difference between a missense mutation and a nonsense mutation is that a missense codes for any other amino acid. A nonsense mutation codes for a stop codon. It basically just cuts off there. Okay. The amazing thing is the difference between FOX genes throughout every animal is generally 1 or 2 amino acids. The difference between a mouse and its speech patterns and almost every other mammalian, no, not even just mammalian, every other sort of animal out there is 1 nucleic acid, and us as humans are 2 nucleic acids different than the great apes, which is crazy, and I love it. And next. Okay, I can't put in a human FOXP2 gene into a mouse and then expect it to be Stuart Little, which is depressing because that would be awesome. Also kind of sad. A mouse bred with absolutely no FOXP2 will have an abnormal squeak. It's usually noted as high-pitched and sort of grating, and very slow cognition, especially like in the controlled experiments that are taking place. The interesting thing is, as soon as you add in the human FOXP2 gene, my squeaking patterns, recognizable patterns, and it's usually a lot deeper, which sort of indicates a physiological difference with not only the mouth, but sort of the voice box and that sort of area. They also have a slightly different brain structure, particularly when it comes to cognition and the way that their sort of thought patterns fire. And as a result, they are faster at learning at least the tests that arise from the cognitive testing. Yeah?
What do you mean by recognizable squeak patterns? Because I figure if you listen to, I mean, I've never particularly listened for an extended amount of time mice, but I figure if you listen to regular mice, you could probably pick up squeak patterns as well.
Probably.
But like, are they distinct from regular mice?
Yes.
There are some studies, and this is almost more anthropology, or zooanthropology, where people have tried to figure out animal equivalents of speech, usually in something with demonstrated degree of intelligence. And you can usually see some basic patterns in most creatures, or some basic types of things which indicate some really, really simple concepts like, over that side there's some fierce predator will fall on this side of the tree.
Yeah.
Or food, I found food!
I think most importantly of all, hey, do you want to have sex?
We can usually map a couple of very
Yeah.
basic things like that.
The thing that I
They
think
call
about
them marmots, so
squeak
they can
patterns is
describe
like our grammatical
Yeah.
patterns.
what they're wearing.
It's notable patterns sort of. They have a sort of cadence and a pattern similar to grammatical sentences that isn't necessarily displayed
new mice weren't understood by controlled mice, like
Yeah,
regular mice.
they weren't at all, which was surprising. And I'd honestly really like to see what happened if two of the human FOXP2 gene mice actually communicated together.
They could take over the world.
They probably could. I mean, it happened in a hitchhiker's guide, and that's totally legit.
Pinky and the brain.
Pinky and the brain.
What is the genius? The
What they
ultimate
didn't say.
the laboratory of the genius has to play. Pinky, okay.
Carry on. Sorry,
Okay.
Okay, this is where we start getting into genetic stuff. So, we're going to start with RNA interference, because that's the stuff I know the most about. Okay.
Just quickly, could you run through exactly what the difference is between genetics and epigenetics?
I was going to get to that afterwards, but I'm happy to do so after. Okay, epigenetics, basically gene expression and regulation of genes that happen outside of the transcription of the, this is what is on the gene, this is what is produced. That's basically just everything else that happens. I have a better wording for it on the slide that's coming up next, so you'll be fine. Okay, so RNA interference. Okay, how do I start? You'll have RNA that is either introduced into the cell or produced naturally by the cell, and this is usually double-stranded. The reason why it's double-stranded is so that this thing called the dicer and argonauts, which is that purple thing there.
They're just random names. They're not really important.
Yeah, they're not important. But the thing is, they cleave the double-stranded RNA in two, then they combine with one half of it, which is known as a template, and then they create this thing called RISC, which is like ribonucleic. It's basically a sort of coding mechanism. It doesn't really matter. Okay, so they bind together. They go around in the cytoplasm, so outside the nucleus, and they go and find any messenger RNA that matches up with the template. Once that happens, they attach to it and they literally cleave it in two. So the RNA that is cleaved disintegrates and it goes off on a basic murderous rampage, going and regulating gene expression by getting rid of all of the messenger RNA that matches up to that specific sequence.
It's basically giving a berserker a picture of the people you want it to kill and saying go.
Yeah, basically. So that's what RNA interference is like. The reason why I bring that up is because not only is it made naturally by some cells, not all, because this has been found in certain nematodes, which are like roundworms and flatworms, and they've been found in fish. But we've also been using them as a form of epigenetic medicine. We're trying to use it as genetic therapy instead of going and trying to get within the nucleus and inserting genetic information like that. We can just put in this double-stranded RNA and basically give the berserker a picture of the stuff we want to take out. And that's basically how it works, which is nice because it's not permanent. It can be sort of removed at any time simply because that complex will eventually break down. And it helps because it's just easier. Trying to insert DNA randomly into the nucleus, into our genetic material, has a lot of dangers associated with it because we are yet to control where on earth that new sequence is placed.
So you get cancer.
Yeah. Cancer or death.
Just plain old death.
Cancer or death. Okay, cake, please.
We're out of cake.
Here is a better explanation of what I was trying to say before. Epigenetics. It means there are changes in phenotype or other gene expression by mechanisms other than changes in the underlying DNA sequence. I will talk about phenotype because I neglected to. Okay, phenotype. DNA, RNA, protein, genotype. To phenotype. Genetic results, actual physical results. Does that make sense? Phenotype is basically this is what it looks like. Genotype is this is what it's made out of. Alright. all those like silly voices. Yeah.
Being like, there's like 10 different ways to make a certain type of eye color.
Yeah.
So there's like
Yeah.
different gene types, and then they all just make brown eyes.
Eye color kind of hinges on the fact that you have melanin, and it's basically you control the amount of melanin that is expressed in the eyes, and you get different eye colors. And I think it was established, or at least there's pretty good theory going around that because the blue-eye gene is technically quite rare among the population, that's the mutation that means that barely any melanin, that one, gets expressed within the eyes, that it came from one family. Which is kind of cool, because we can sort of trace it back.
People are inbred.
There are now studies that show that you can skip the melanin from the eye if you give someone who has brown eyes blue
Yeah,
eyes.
you can do that now, which is kind of weird. So I wouldn't like chemicals that strip melanin to be put on the eye.
It's one of those, like it happens over the course of a week, I think,
Yeah.
and then melanin slowly disappears from your irises.
Yeah. So in that case, it's a phenotypic change. It's just chemicals that can be unreal. Whether I want to change brown
I
eyes
think
blue.
it's that you have to have the blue eye gene already in your, like you have to, like have the possibility of having blue eyes first, so you can't just chuck an Asian in and say you can have blue eyes now.
Unless they have blue eyes.
They probably be the biggest market.
Which is possible.
Anyway.
All right. On that note, like just while we're on the blue-eyed gene thing, I'll say it because blue-eyed genes is a recessive trait, whereas brown-eyed is usually a dominant. Dominant and recessive. We're not going into much Mendelian genetics at the moment. If you want to learn about it, Google or Wiki Gregor Mendel and his pea plant experiments, because it basically goes into, what certain traits of the gene are dominant and what are recessive. It's certain patterns of dominant and recessive genes bind together, what would their expected progeny be, or expected children be. So if you want to look it up, that's sort of more population-based, because they're sort of ratios of, how many children will have brown eyes compared to blue eyes, or how many, or something like that. So I'd recommend looking that up if you want to learn more about, how dominant and recessive genes work in a small controlled environment, such as pea plants. Okay. Now we're going to look into two methods of epigenetic regulation at the moment. The first one is methylation, and the second one is chromatin modeling. Methylation time! Alright. Methyl groups, so just CH3, bind to a cytosine base connected to a guanine. This is known as a CPG island. Yeah, a CPG island, and a methyl group will usually bind to that, as you can see right here. This results in basically gene silencing, because when the complex comes in and tries to transcribe the DNA to RNA, it's literally blocked. It can't get through that methyl group. It's like a wombat getting in the middle of the road, basically, and you just try and ride past it, but no, it's a boulder with legs.
You can get past it on that. You can't.
Yeah. So basically, it can try and get past it, but there won't be any RNA or any functional RNA that would be produced in this.
Your car is crashed.
Yeah, basically.
All
So,
crumbled.
cytosine basically looks like it without being a methyl group. So this is what cytosine looks like. It's known as a purine. The pyrimidine has two, doesn't it? Yeah, so this is the purine, and it would join up with G normally. As soon as you get that CH3 in, the C and the G bind okay, but the sugar phosphate backbone gets warped, and that's mainly what prevents it from going along the DNA and transcribing it. It can be inherited, which is a cool thing, if it's methylated in gamete cells, so in sex cells. If this can was methylated, they wouldn't get it, basically. Okay, so the way it works is that when DNA gets replicated, after it's copied along, DNA methyltransferase will go along the template strand and basically be like, ooh, there's a methyl group here, I'll add one to this side. Because not all methylations are bad. They aid in a lot of things, and they can hinder a lot of things. But the fact that epigenetic regulation can be inherited, but also can be changed outside of development. So if you were to eat a lot of methyl-rich foods, you could technically get methylation. It's something to keep in mind, especially when you're, on a diet or expecting a child or whatever.
So, this is an environmental influence which is inherited.
Yes.
Yes.
So that means Lamarck was not entirely wrong.
Yes.
Basically, the more we learn about genetics, the more we're kind of like, oh, so we were wrong about that.
Yeah, basically, the more we learn, the more we think that every theory sort of has some little bit of merit.
If you apply it to this teeny tiny section.
Yeah, you apply it to a little bit. So Lamarck was wrong in terms of, this is solely how you evolve, but Lamarck was right in the sense that outside factors do have slightly influence.
Just quickly, Lamarck was a biologist who basically thought that things that you inherited during your life could be passed on to your children. Everyone thought he was an idiot for years and years and years, and then we discovered this.
Yeah, and they were like, oh, we're the idiots. Right.
So it's like a theory that if you learn to write, because you've learned that, your children will learn to write.
Well, it was more with giraffes.
With giraffes
There's one example that they'd be stretching, and then through their stretching, the kids would have
Yeah.
longer necks, and over generations of stretching.
Certainly not on such a large scale as that. Certainly not. It's mainly the methylation. What examples can I give for that?
The
The
bees.
thinking depression.
Yep.
What kind of things have a lot of methyl in them?
Methylated spirits.
Yeah,
Pretty
don't
much
drink
a
that.
lot. Basically, any food you think of as healthy is probably got a lot of methyl in it.
Yeah,
Everything they recommend you to eat during pregnancy is basically methyl rich because that methylation is part of one of the things that prevents things like spina bifida. Is that correct?
yeah. So lots of leafy greens. Salmon is also incredibly good. Less processed food generally, although that can be applied to anything. So just generally eat less processed food because fresh food is tasty food and stuff. What was I going on about? Oh, the Hungary depression thing. This is just ad-lib, so this might not be entirely, it's going to be somewhat correct, but it might not be entirely actually incorrect. Look it up. Because Hungary at the time kept going into bouts of depression and people would get malnourished.
This is the country Hungary?
Yeah. And people they got invaded again and again and again. They got into periods of depression. They had, financial depression and of course a lot of families would be pretty down because their country kept getting invaded. Basically what happened is they'd go through periods of massive sort of food shortages and they'd get they'd sort of binge eat to sort of get to sort of regain what they had previously lost. They found out that their children were less likely to develop obesity. They'd become really long and they'd be really slender, and this was noted in most offspring of the people from those generations. And because it kept happening again and again and again, there wasn't enough time for it to sort of go back to its normal state. And I think one of the notable figures that was given as an example for this Hungary depression sort of methylation was Breakfast at Tiffany's, that girl.
Audrey
Hepburn.
Hepburn. No, she was the Dutch famine victim. But yeah, same thing.
Same thing, but it was the Dutch famine victim, basically. Audrey Hepburn is a notable example because they found that they had this methylation pattern that corresponded with the people from Hungary. Let's keep going. Okay, so remember how I talked about DNA flying around these things called histone proteins? Histones are kind of cool as they're sort of full like ball structures with heads on them, and they sort of act like massive balls of Velcro. And whenever they can, once DNA winds around the spools, these little hairs sort of latch onto them and sort of keep them in place. But the thing is, they can be acetylated, which is happening here, and they can also be methylated, which you can see here. And once that happens, they're sort of just waving around everywhere. And the outcome of that is that these can unwind. And we've already talked about the implications of unwinding. They're at higher chance of mutation, which is not very good. The difference is these are far less likely to be inherited even. But yeah, basically this epigenetic regulation is non-specific. It's just the outcome of this is a far higher chance of mutation. What next? Oh yeah, super question bonus round. Yay! What do people want to talk about or want me to rant on about, or what do you think is cool that you want to share with people in regards to
Tell us
genetics?
about your mutation. Because
Yeah,
we're worried.
okay, I'm gonna draw pretty pictures. Just like I'm drawing pretty pictures. Ladies, you're not allowed to use the bathroom.
We're aware.
No, just look at
Awesome, please go. Okay. I have something called a 4-5 translocation. Does anyone know what a translocation mutation is? Apart from you two.
A chunk of your chromosome is in the wrong spot.
Yeah, basically, I have something called, 2-4 translocation. So let's say this is chromosome 2,
So is it a crossing over thing?
She's about to explain
and
it.
this is chromosome 4.
looks like a bunny.
It kind of does. Also, they're really messy and not at all accurate. Well, we'll get into that. It's the messy. Okay. Next. This part of chromosome 2 loses its top.
It danced too hard and its arm fell off.
Basically. This one got a new hat. That is basically what a translocation is. The difference between a regular translocation and my translocation is that it's balanced, which means it's cut, luckily, where a gene wasn't. That's the whole reason why I'm surviving, guys. Cut where a gene wasn't and attached itself where it wouldn't destroy anything else.
So you won the genetic lottery.
Yeah, basically. So did my dad. Terrible. My dad and my granddad have it. Yeah.
You only have to play the lottery once. You didn't actually have to play.
They have to play the lottery. I just reaped some of the rewards.
What does that do?
Pardon?
What does that do?
Well, I may or may not be Wolverine. In all seriousness, it does absolutely nothing that I know of.
Basically, one DNA repair mechanism is that if part of one of your chromosomes becomes detached for some reason, it'll just reattach it wherever it can, because there is a chance that even if it reattaches it in the wrong place, that at least means the genes on the detached chunk won't be destroyed.
Yeah,
And they
they'll
can
still
be
be there and they'll still technically be functional.
They're just in the wrong
One thing
spot.
I also will add is another repair mechanism and something called telomere onto the end of it. So that way, because what happens when DNA replicates, the cell replication, is that it always misses a bit of the end. But you don't want that bit of the end to be an actual gene, so they just add in junk, basically. They just add in,
It's a repeated sequence.
yeah, a repeated sequence of like 6 or 7 or something like that. A repeated sequence like thousands and thousands of times. So that basically no actual content is lost.
White noise.
Yeah, pretty much. It's white noise to protect your genes.
Yeah, basically. So that's my mutation, and it's really cool. But I'm not special because everyone has at least like several hundred point mutations in them. Mine's just a little more.
Impressive, since it's a chromosome.
Yeah.
And we don't have many of those.
Yeah, that live anyway. I ended up photocopying the medical certificate from my birth, and I tacked it up onto my wall just because I thought it was cool and that I could maybe possibly Wolverine and stuff like that.
Did they do karyotype?
A karyotype, yeah, they ended up having to do a karyotype of my dad and me after that, just to check that I would be a viable organism. But it's basically just balanced to 5 basic X 30 something XX. My brain is fast. It's just a normal karyotype that just mentions that it's
46, by the way. With 36, you'd have a serious problem.
36 is important.
No, that just means you're
chromosomes you have.
you're missing 5 chromosome pairs.
My father has like a hammer toe, like, you know, the ones where it's kind of like the pinky's like bigger and bent with the hammer.
No.
Mutation.
Yeah.
Oh yeah.
That thing.
allele is basically just one certain mutation of a gene that's viable.
There's some evidence against that now, but
Hmm.
it's a good example. Better example of that is whether your earlobes are attached
Yeah.
or not. Like mine are attached, there's no fold under them, whereas say Grisha's hang down a bit, there's a little bit of detachment.
The ears hang low, do they wobble to
He has loads.
It's the same
Earlobes, whereas mine
Right,
aren't, well, mine are attached.
Yeah.
I don't obvious that like don't
Yeah.
we just go straight in.
That's more because I spent years and years wearing heavy earrings.
Yeah.
What's
Okay.
the deal with web feet? Like-
Go back one.
Oh, sorry.
No, you go.
Well, what's the deal with web feet? Is that like an actual carryover from organisms that did have webbed feet? Or is it just like your feet are webbed because of a random mutation? And not like a really recessive?
In our case, it would be a random mutation, just because we evolved differently to something like that, which do have webbed feet. However, there are.
So we just never had webbed feet.
I'm fitting instead.
Our
Nothing
ancient ancestors probably
It wouldn't be conserved for that long.
Yeah,
Exactly.
basically. Like, in order for us to have webbed feet, we'd have to have a mutation in the Hox genes that change that.
The same as the decidual tail, isn't it?
Yeah.
Yep.
It's been out of humans for a long time. We can't remember that.
Well, I mean, if you look at apes, that's basically how far
Mhm.
gone it's been.
Even the safe don't have.
Exactly, well,
Apes
apes don't have
don't
apes.
have
That's
apes.
part of the feature.
Just got another question. We mentioned blue eyes before. So, like, if they all came from one family, do we have any evidence as to why blue eyes became so much more predominant around a wider human population?
It's
And
basically
the
a
human
numbers
population-
game.
It's a
And the
numbers
human
game.
population was very small at a couple of places in the street.
It doesn't even come into it. I mean, Mendelian genetics shows that basically because it's what we call a recessive trait, it kind of hides in the background.
Sure.
So a lot of people may not have had blue eyes for that generation, but they had one of their genes was the blue-eyed gene. So when they had children, there was a chance that one of their children would have blue eyes, and all the other ones might have a little sneaky blue eye.
Yeah, they
gene.
might be carrying it, but not.
So it
Yeah.
spread secretly, and you wouldn't really know.
I'm gonna
Well,
draw
what I mean
it
is,
flat.
like, is there an evolutionary advantage towards blue eyes?
There's certainly, well, yes and no.
Well, I mean,
Humans-
the cavemen think, wow, blue eyes.
Previously thought, basically the rarer the thing, the more they wanted to get it on. And blue eyes at a point were really rare. Yeah.
And there are distinct genetic advantages in certain parts of the world to having much less pigment.
Yeah.
Sure,
If you take a black person up into the Antarctic or down into the Antarctic, they're going to get huge issues with not enough vitamin D. You take Sarah and put her in the Sahara, she's going to crisp.
Yeah. I mean, that's part of the reason particular colourings evolved in particular parts of the
No,
world.
well, I was about to say,
There's
no,
some
that's why
evidence
it's
that
towards
also
Johnny.
blue eyes will be correlated to high alcohol tolerance.
Except for
Not in
vodka.
my case.
I can prove this wrong.
Exactly.
Get
Not
me
in a
drunk.
scientifically
You'll take
rigorous
a
way. You
one
cannot
wine.
prove this wrong. Your science properly.
I don't know, it depends how big a budget I have.
I think you're saying anecdotally her evidence will prove us wrong.
No.
Use science.
That's why I need a budget.
Alright, understood. And the alcohol.
I'm pretty sure you're the same for alcohol.
Don't you want to make one of those?
I'll get the participants.
I'll participate. Although you probably want people without alcohol.
That'll
That
do.
would be quite hard to find.
Anyway, cool shit.
Okay.
Look at the
This
cool shit.
helps.
Look at the planets and the squares.
This helps with like the patterns of inheritance, particularly with blue eyes. Okay, let's say the dominant brown eye gene is the capital, and the recessive blue eye gene is the lowercase. Okay, so Gregor Mendel, when he did his plant experiment, noticed this in like real life from breeding and then breeding again and cross breeding.
Is that where they get the name because he used punnets?
Ooh.
We've never been taught that because nobody
I'm sure
really
you
gives
can
it
make
to
a
us.
fun out.
I give it, yeah.
Okay, so
It's ratio count, you're not important.
having, let's start with down. okay. Having one chromosome with two of the same allele on it, so two of the exact same copy, are known as homozygous. When two of them breed together, all of their offspring are homozygous.
And basically, you have to have two little bees to have the blue-eyed, to be blue-eyed. If you've got even just one big bee, you're going to have brown eyes.
Yeah, because it's recessive. This one like fades into the background because the dominant one is so obnoxious it can't help it. It's like, look at me! I'm the brown eye!
Even though brown.
You see, like what? Yeah.
I don't know. I don't like the color brown, so.
But the thing is, you would if it was red.
Yeah.
No, I don't like the color brown at anything really.
No, but that's the thing. If it were rare, it'd be kind of exotic.
You'd be genetically attracted to it.
It's okay.
I haven't seen that before.
To further demonstrate the whole dominance
Genetically, yes.
thing, if we were to combine two heterozygous, so if they had if they were to have if they were to have two different alleles, both of them, they're known as heterozygous, you get one which only expresses the dominant and only carries the dominant. You have two which express the dominant but carry the recessive blue-eyed gene, and you have one which has the blue eyes, and it's known as the 1 to 2 to 1 genotypic ratio, or the 3 to 1 phenotypic ratio. Does that make sense?
Yes.
Okay, and sort of same case here. If you were to have homozygous recessive and homozygous dominant, in this case, all of them would carry the silent mutation, not silent, the recessive gene, but express the dominant. Anyone got any questions? Anyone want to add more cool things to this discussion? Because I like talking about cool things. Or want to ask any questions? Because I'm happy to answer.
Don't get into how you get green eyes, it's not worth it.
Oh my god,
Green eyes.
green eyes are a genetic clusterfuck.
Yes.
It's very offensive though.
Awesome.
My genetic clusterfuck is pretty.
My eyes
Yeah.
change colour, you don't even go there.
It's sort of, yeah. Eye color, eye color is a bad example to use for this, because there are many, many, many different factors, apart from just genetic traits. That affect
eyes.
Yeah.
That's pretty much.
Yeah. What's really funny is my dad has brown eyes, my mom has blue eyes, but one of my siblings had blue eyes. But I've got green brownie eyes, and my sister has green eyes.
What colour eyes do they have?
Did you check the milkman? Your sister having blue eyes is somewhat expected, somewhat. Green eyes, again, it's sort of a mix between this is what patterns of inheritance are, and then this is what happens epigenetically.
Because we found out later that my father is a brown blue,
Yeah.
and my mom's a blue blue.
Yep.
So I know I've got like a blue in me somewhere.
Black don't color.
Pretty much. But then I ended up with green, and
Are you
everyone was like,
like
fuck.
What did you do with the blue?
Okay.
Okay. Anyone else want to say cool things, or shall I start demonstrating?
I think you should start demonstrating.
All right. I'm going to pass this around.